All right, we're going to talk about evolution and populations. All right, when we say the word evolution, we're going to talk about evolution first. We talk about Charles Darwin. Charles Darwin is the guy who is responsible for coming up with proposing the theory of evolution. When we say theory, that means it's been proven over and over in science. And the way that he proposed the theory of evolution is he said it occurs by natural selection. Now, this was in the 1800s, and there's a lot of criticism about evolution. So the way Charles Darwin basically explained natural selection is he talked about in the 1800s how they were breeding dogs and getting desired traits. So that's what we know as artificial selection. So natural selection we'll talk about, but think about it as in terms if you want to make a new designer dog, you would use something like natural selection, but you're artificially, taking two dogs and you're breeding them. So Charles Darwin is known as the father of evolution. And I'm going to give you a little back story on him. You should take some notes. I'm not going to write everything else out. Okay, Charles Darwin was a naturalist. He was a naturalist. I'll write that out. And his job is he got hired on a ship called the HMS Beagle in 1831. So what his job is they sailed around. They were in a ship, and his job as a naturalist was to basically just observe everything that he saw while they were traveling, around the Galapagos Islands and the different places that they went. I believe it's a Spanish ship. And not only were they, recording what they saw, but they were also basically making maps, graphing out everything. They were seeing where everything was, trying to claim new land, find new land. So he was on the HMS Beagle. He was a naturalist, and he was writing about all the different things that he saw. And then when they went ashore anywhere, he would collect bones, he would collect fossils, he would collect rocks, he would even collect animals, skeletons of animals, all kinds of things. That's what his job was. And over like a span of 20 years, he basically was thinking about what he saw, and he basically came up with this idea that some of these birds, finches and the Galapagos tortoises, he basically was saying some of them were adapted. Some of them had a different appearance to them. So he studied finches, like a bunch of different birds on the Galapagos Islands. Here's a little bird. I'm going to try to draw a bird. I'm not very good at drawing birds, so bear with me. And he noticed that some of the birds, had very large beaks, and some of them had very small beaks. And so he was like, some of the birds are the same exact birds. They look different. So he said, well, okay, they have inherited characteristics that help them live. So that's what we call an adaptation. So these are inherited characteristics. Of an organism. That enhances their survival in an environment, and reproduction. So finches, you know, they eat seeds and things like that. So he noticed that, you know, ones with small beaks had an easier time, you know, eating small seeds, and the ones with very large beaks, let's make this one a little bigger. It's a big nose. Ones with large beaks versus small beaks could live on bigger seeds. So if you were born with a larger beak and there were an abundant amount of large seeds on the Galapagos Island, then you would live. So you would survive better, you're going to reproduce more. And that's what he noticed with these birds. So he wrote a book, and his book is On the Origin of Species. I don't remember the year. Let's see. I know it's like 20 years. I'm trying to remember. Oh well, let's just write it on. I'll maybe remember it. So he noticed all these adaptations. So his book, after reviewing everything that he collected on the HMS Beagle, he wrote the book On the Origin of Species. By means of natural selection. This is his book. Oh, it was 1859. I knew it was 1850 something. I just didn't remember. So, in 1831, he recorded everything. So more than 20 years, he was like just thinking about everything he saw. I'm guessing he looked at all the things that he collected, and he was like, where are these adaptations coming from? So, in his book, he said, well, adaptations come from natural selection. That's what he listed. So we're about to talk about natural selection. Adaptation arises from natural selection. Now, when you say natural selection's definition sounds exactly same as adaptation. However, this is going to be over years. It's a process. So overnight, you can't just wake up and everybody's going to have big beaks. This is going to take years and years and years. So natural selection is a process. It's a process. So in nature, we're going to pass down inherited characteristics that helped the previous ancestors live and survive. So natural selection is literally a process which an individual with inherited traits tends to survive and produce at higher rates, and it's going to be passed to their offspring. So, evolution. Let me first say, Darwin did not use the word evolution, not one bit. He said the word descent with modification. So, even though we call him the father of evolution, that is not his word. What Charles Darwin said was descent with modification. In his book, he said evolution is descent with modification. So when we say evolution, this is the idea that living organisms or living species are descendants of ancestor species, because it's descent with modifications, modified over generations. And the way that we've modified over these generations is natural selection. This process where we've adapted. We've adapted over and over and over by means of natural selection. The process is natural selection of how we modified. So when we say that we're going to discuss evolution and populations, basically we can look at if there's a change in a population to see if evolution has occurred. So when we have a change in any of our genetic makeup in a population, then we know that evolution has occurred. So that's why we're covering evolution and populations. Now, evolution is going to basically be change in the genetics in a population because it's over generations. Change in genetics in a population is going to help us measure evolution. So we have something called population genetics. This is what we're going to discuss, population genetics. Before we go over population genetics and how to calculate if evolution is occurring in a population, we need to go through some definitions. So, you should definitely review your genetics because we're going to be using the words homozygous dominant. You should know what homozygous recessive is and heterozygous because we're looking at population genetics. So population genetics is the study of genes. So we're looking at how many homozygous dominant individuals are in a population. How many recessive people there are, and how many heterozygous people are. And when we say studying genes, we're studying the genes in a population to see if evolution is occurring. Now, one of the smallest terms of evolution is called microevolution. Let me change colors. This is going to be like a subcategory. Microevolution. When you have a change in allele frequency, so we talked about dominant alleles. So you have a dominant allele versus a recessive allele. When you have a change in the number of alleles in a population over generations, that's microevolution. So we can use population genetics to see if microevolution occurs. So just because you have a change in allele frequencies doesn't mean that you have a full-on change of your phenotype. You can just have a different number of alleles. So over time, you go from having a bunch of big beak finches to now all of a sudden they all are starting to have short beaks, but changing the alleles. So when we study population genetics, what we have to look at, since we're studying genes in a population, is we have to look at genotypic, to study population genetics, we study genotypic frequencies are going to be the percentage of, or not percentage, well, it is percentage, the percentage of homozygous dominant, heterozygous, and homozygous recessive individuals in a population. So to study population genetics, we're going to study genotypic frequencies. So I want to know how many homozygous dominant people there are in a population. What's the percentage of heterozygous individuals in a population? And the percentage of homozygous recessive in individuals. And then the next thing is that we study allele frequencies, because allele frequencies are going to lead to changes, they're going to lead to microevolution. So allele frequencies is, I want to know in a population the percentage of dominant alleles versus recessive alleles. So this is how we study population genetics. So the next topic is over genotypic and allele frequencies. Now, I'm not going to start out with the Hardy-Weinberg equations just yet, but I do need to cover it. When we study population genetics, we use two equations known as the Hardy-Weinberg equations. This is what we use to predict whether a population is evolving. So these are two scientists, and we are going to be doing Hardy-Weinberg. So to predict if a population is evolving, what we use are Hardy-Weinberg equations. These are going to be very important. Now, most of the time when a population is not evolving, your allele frequencies are always going to add up to 1, and your genotypic frequencies are always going to add up to 1. So, Hardy-Weinberg equations, there's two of them, which we're going to cover later. P plus Q equals 1. P squared plus 2PQ plus Q squared equals 1. These are the equations that we use to predict if a population is evolving. We'll come back to that. Now, we're talking about population genetics. This is population genetics. So we need to understand a couple of definitions before we move on. Definitions that you need to understand. What is a population? So a population is a group of organisms. So we could talk about Big Spring, Texas. We're a group of organisms. Humans specifically belonging to one species in a certain area. Now, the definition of a population is a group of organisms belonging to one species. So here in Big Spring, we have a population of horny toads, and we have a population of humans. So we can actually determine if evolution is occurring in horny toads by using the Hardy-Weinberg equations. But when we look at the Hardy-Weinberg equations, we have to know our genotypic amounts. We have to know how many homozygous dominant there are. We have to know how many heterozygous individuals there are, how many homozygous recessive individuals there are to figure out the genotypic frequency. So, if we're talking about population genetics, then we're going to look at horny toads in Big Spring, Texas. So if that was the case, then we would have to catch all the horny toads in Big Spring, and we'd have to start testing them so that we know what our gene pool is. So, a gene pool. Is the number of alleles in a population. We need to know exactly how many horny toads there are, and we need to know the number of alleles. So, in population genetics, when we say allele frequency, I want to know the frequency of the dominant allele versus the recessive allele. How many dominant alleles versus recessive alleles in a population. So, big B versus little b. Allele frequencies. So there's only going to ever be 2 alleles that we're going to work with. The next thing that we look at is genotypic frequencies. I want to know how many, I'm not going to write this out, homozygous dominant individuals are there versus heterozygous individuals versus homozygous recessive individuals in a population. So, before you can figure out if a population is evolving, you have to figure out the number of organisms of that one species in a certain area to figure out the frequency. When we use the word frequency, we're using the portion of individuals in a certain category. So when we talk about genotypic frequencies, I can figure out the number of homozygous recessive genotypic frequency. I can figure out the number of individuals with this heterozygous and determine how many percent are in a population. Now we're going to use 2 equations for these. Now, the first one, there's going to be equations. These you need to understand. For population genetics. So, genotypic frequencies. Frequency is the portion of an individual in a certain category. When you say genotypic frequency, we're trying to figure out the frequency of homozygous dominant versus heterozygous versus homozygous recessive. So, frequency is the portion. To figure this out, you take the number of the particular genotype, so it could be homozygous dominant, and you divide it by the number of individuals in the population. That's the total number of individuals in a population. Now we're going to do some practices on this, but you have to understand these equations for the lab practical because you're going to have examples in your lab that you need to work through or your worksheet, and you're also going to have these that you need to understand them, like in your post lab or your worksheet. So your allele frequency. Now these are used for a population. So to figure out allele frequency in a population, again, we're trying to determine the number of dominant alleles versus the number of recessive alleles. So the number of particular alleles, whether it be dominant or recessive, divided by the number of alleles in a population. Now, everybody has 2 alleles. Your genotype is 2 alleles. You either have 2 recessive alleles or a dominant and recessive allele, or 2 dominant alleles. So to figure out the number of alleles in a population is you take your total population. So you take your population size times 2, because everybody has 2 alleles. That's how you figure out the number of alleles in the population. So I have an example for you. We're going to come back to these. So we're collecting horny toads. We know that the population of horny toads is 12. That's just my example. I have 12 individuals in a population of horny toads in Big Spring, Texas. And I tested all of these. So, of the 12, 5 of the horny toads were homozygous dominant, and 5 of the horny toads were heterozygous. So that's 5, 10 people. So that means I only had 2 that were homozygous recessive. So that's my total population. Again, a population is a group of individuals of one species in a certain area. So, just saying 12 horny toads in Big Spring, Texas. So, you can figure out first your genotypic frequencies. So, I want to know the genotypic frequency of homozygous dominant. So you use your equation. It's the total of a particular genotype divided by the total number in the population. So I look at this and I say I have 5 homozygous dominant. So to figure out the frequency of homozygous dominant genotype, remember, genotype, frequency, is equal to the number of a particular genotype. So this is particular. Divide it by the total population size. So how many horny toads did we have? We had 12. That's my population size. And how many homozygous dominant genotypes did I have? I had 5. So you take 5 and you divide it by the total, 12. Now, one thing I need to write on here, and I'm going to write this in red. Frequencies are always written as decimals. So for the purposes of turning in any work, I only want you to use 2 decimal places. So we can take 5 and divide it by 12, and it should be a decimal. I'm using my phone to record. Let me pull up my apps. You can find a calculator. You're going to need a calculator for this. You can use your cell phone. All right, let me launch I pulled up my computer. Took me a minute to get it up. I had to log in. All right, 5. I'm sure y'all have already figured it out by now. 5 divided by 12 is 4.416. So we're only going to use 2 decimal points. So you would say the answer is the genotypic frequency of homozygous dominant individuals in a population is 0.42. That is your answer. So you should be able to do genotypic frequencies for each of your genotypes. So the second question, figure out your genotypic frequency of heterozygous individuals in a population. So again, the number of a particular genotype, which is given, there's 5. The total individuals is 12. So again, the answer is 0.42. So if asked, what is the genotypic frequency of heterozygous individuals in this population of horny toads, it's 0.42. Well, since we have 3 genotypes, you go ahead and you do the third one. I want to know the genotypic frequency of homozygous recessive genotypes in my population of horny toads. So again, my particular number of particular genotypes is 2. I only have 2 of them. Divide it by the total population. We're going to put it in 2 decimal points. 0.166666, so is 0.17. Now, usually in a population, we use Hardy-Weinberg equations. And Hardy-Weinberg equations are always going to be equal to 1. So knowing the number of populations, when I try to figure out genotypic frequencies, I should be adding up these 3 because there's 3 different genotypes, and it should equal 1 or close to 1. So 0.42 plus 0.42 plus 0.17 is equal to 1.01. So we're always going to write our decimals in 2 places. 1, 2. 2 places as our answer. So Hardy-Weinberg would have calculated the same thing when doing genotypic frequencies using their equations. We have 3 genotypes. Hardy-Weinberg equations say that these should equal 1 unless a population is evolving, which it is not because it's equal to 1. So the next thing is you need to figure out your allele frequencies. Now, allele frequencies are a little different. You are given a population, but you have to figure out the number of total alleles there are. So, to figure out allele frequencies, you're going to go through a couple of steps. So the first thing that we did is we used the information given to figure out genotypic frequencies. Well, we're going to use this same information. Everybody carries 2 alleles. You're either homozygous dominant, or you're either homozygous recessive, or you're heterozygous. So the first thing, because allele frequencies is the number of a particular allele, so this is going to be, since we're using B versus little b, I want to know the number of dominant alleles versus the number of recessive alleles. We need to know the formula. The allele frequency is the number of a particular allele. There's only 2. There's dominant allele versus recessive allele, divided by the total number of alleles in the population. So basically everybody has 2 alleles, so you take your population size times 2 to figure out the total number of alleles. So if we have 12 individuals, everybody has 2 alleles. We say 12 times 2, the number of alleles in this population would say 12 times 2. There are a total of 24 alleles in the population. Now, to figure out how many alleles there are, you have to look at your genotypes. So, you have 5 individuals that are homozygous dominant. So they have 2 dominant alleles. So, for the dominant allele, I need to figure out how many dominant alleles are there in this population. So, since you have 5 homozygous dominant, that would mean you have 10 big B alleles from your homozygous dominant, and you had 5 that are heterozygous. So when there are 5 that's heterozygous, that means they have a big allele plus a little allele, so I'm only going to count half. So this one, to figure out allele frequencies, I say 5 times 2, because there's 2 of them. This one, there's only 1 set. So, plus 5 big B's from heterozygous. So I have a total of 15 dominant alleles. That is going to help me for the next step. Now, the number of recessive alleles. I have to figure this out. Now, before I move on, this and this number should equal 24. So I go back to my original thing at the top. I have 2 homozygous recessive when they have 2 little b, so 2 times 2 is 4. So I have 4 little b's from this one, but I also have 5 little b's from this, so I have plus 5 little b's from the heterozygous. I add those up, and I have 9 little b's. So in the population, I have a total of 24. So if I add 15 plus 9, that equals 24, so I can move on to the next step. So for allele frequencies, we want to know the number of dominant alleles versus the number of recessive alleles. So we're on E, allele frequency. What is the allele frequency of the dominant allele? So back to our equation. How many dominant alleles do we have? So we had 15 dominant alleles, which we calculated. So we would say 15. It's the number of the particular alleles divided by the total number of alleles in the population, which we calculated here. There are 24 total alleles, so we take 15 divided by 24, and it's a frequency, so it should be in 2. 0.625, so we're going to round to 0.63. The next one, what is the allele frequency of the recessive allele? Little b. So go back to my 24 alleles is the total, but I had 9 that were recessive alleles. So 9 divided by 24 is 0.375. So 0.38. Now, since E and F are both allele frequencies, you should be able to add up 0.63 and 0.38, and it should equal close to 1, because Weinberg says it's always 1. So 0.63 plus 0.38 is equal to 1.01. Now your decimals, we're just keeping them at 2. If it rounds up to if it goes to 0.99, don't worry about it as long as you do your decimals right. So allele frequencies, there's only 2 alleles. You have your dominant allele versus your recessive allele. So you're only going to have 2 allele frequencies that should equal 1. But for genotypic frequencies, you have 3 genotypes, so you have to have 3 frequencies, and those should equal 1. So this is an example. This is where I'm going to stop, and you should use your worksheet to figure out your genotypic and allele frequencies on 70 individuals for the first question, and I think it's 47 individuals on the second question by using these examples. So I gave you an example, and you should be able to add up A, B, and C, and they should equal 1. D, you always double, and then E and F should also add up to 1, and that's like a way you double check yourself to see if you're getting them right.